Hey students, hi dear students, welcome you all on the behalf of Rahul Anand Tutorials. So as you know that I am Rahul Anand and this is somewhat on my channel whose name is actually the Rahul Anand Tutorials. So now let's begin with the very interesting topics which are often being asked in terms of your Neet, often the Aiims, often the All India Pre-Medical Examination, as well as what often in terms of the Aiims etc. So these are literally belonging from which of the chapters of your book. So these are belonging from the cell, the unit of life. So today we are going to study about what? So today we are going to study about the endoplasmic reticulum. So when we are talking about the endoplasmic reticulum, you have to go with reference to the two names, such as what? Such as the KL. So your KL Plade as well as what? As well as the Aiims in terms of what? In terms of the 1961 discovered as well as coined what? So they discovered as well as coined the endoplasmic reticulum. Fine. In terms of what? So they particularly performed the series of experiment as well as on the presence as well as they particularly found this endoplasmic reticulum with reference to what of the cells. So it was actually the liver cells. So you are very clear about that. Means what are actually the livers, often playing important role in order to secrete the different types of the cell substances as well as what? As well as these are often having the several types of the vital role in each cell. So we'll be understanding about that in terms of our next lecture. So now let's proceed with reference to the another main important facts in it. So often when we are talking about these cases in terms of what? In terms of the endoplasmic reticulum. So these are what? So these are the site of the protein synthesis. Often the site of the here in terms of the membrane. So you are very clear about that. Means what are actually the protein. So fine. So protein are what? So protein are the macromolecules of the amino acid. Often we are having the four broad kinds of the protein. So these are what? So these are the primary structure, 1 degree protein, the secondary structure, secondary protein, the tertiary structure, 3 degree protein, then the quaternary structure. Remember about the case of the 3 degree structure, often being further divided into the two broad categories. So these are actually what? So these are the fibrous protein, then the globular protein. Often when we are talking about the case of the membrane, so you are very clear. So membrane will be what? The particular portion playing important role to separate what? To separate the ingredients with reference from the external environment or either from the internal environment, or playing important role as like the barrier of what? In terms of the cell. In order for what? In order to protect as well as give it the specialized shape as well as size. Remember about the case of the plasma membrane. So in the case of the plasma membrane, we are having what case? So it will be found where? In terms of the plant cell. On the other side, we are having the leg in terms of what? In terms of the animal cell. Often when this particularly plant cell in terms of what? Which is having the resemblance of the here, I mean the plasma membrane. When it combines with the lamella, it results in the formation of what? It results in the formation of the skeleton of the plants. Now, so when we are talking about the structure with reference to these, means what are actually the resemblance of the plasma membrane. So this will be what? So these are quite a bit round as well as what? As well as oblong. Then what? As well as the tiny tubular structure. So you are very clear about that. Means what are actually the tiny tubular structure. Often we are having the liver, and in these particularly liver cells, we will be having the organelles. So these are what? So these are actually coming under the cell organelles. Fine. Mean in terms of the what? Mean in terms of these. In terms of the endoplasmic reticulum, we are often having the different types of the broad kind of what? The broad kind of the cell organelles. Remember? So you are having the nucleus. Then the nucleus. Role of the nucleus is what? So these are containing the genetic material. In terms of when we are talking about the case of the nucleolus, so these will be having as well as particularly composed of what things. So these are particularly being composed with reference to the RBC. Mean in terms of what? The arteriocytes as well as the protein case. Often you will be having the in different types of the organelles. In term we will be having the here in the what? The endoplasmic reticulum. Often the rough endoplasmic reticulum as well as what? Here many times the Golgi complex as well as the what? Golgi apparatus as well as the vacuole. So we'll be understanding about that later. Now, so when we are talking about often when we are talking about the case of the endoplasmic reticulum, so these are what? So these are the complex network of the membrane bounded with reference to what? With reference to the tubular structure which will be found in terms of what? Which will be found in terms of the electron microscope. You are often very clear about that. Means what are actually the electron microscope. So these are quite bit what? Vital, playing important role as well as having the influence. It is having the major types of the consequence often such as the eyepiece. Often you will be having the lens in terms of the concave lens as well as the convex lens, playing important role to magnify the particular image, which are particularly what? Invisible with reference to the human or either the human naked eyes. Now, when we are talking about the different types of the what? The components which are generally present in terms of what? In terms of the endoplasmic reticulum. So particularly we will be zooming into what? The cell. Then we will be zooming into what? Then we will be zooming into the cell organelles. And after that we will be getting what? So after that, we'll be getting the endoplasmic reticulum. So these are particularly resembling the different types of what? So these are resembling the different types of the parts in it, such as what? Such as we'll be having the nuclear pore. Then you will be having the nuclear cells as well as the nuclear membrane. Often you will be having what? Often you will be having the nucleus as well as what? As well as the rough endoplasmic reticulum. Then the smooth endoplasmic reticulum. Often you will be having the ribosomes. Often when we are talking about the endoplasmic reticulum, so these are known as what? So these are defined as the interconnected network of the membrane bounded tubular structure, which are particularly being viewed as well as particularly being scattered with reference to what? Which are particularly being scattered with reference to your cytoplasm towards what? Towards the particular cell as well as the different organelles as well as the components of what? As well as the different components as well as the organelles of the cell. Now, so when we are talking about these cases, what are actually the cytoplasm? So you are very clear about that. Means what are actually the cytoplasm? Tell me. So these are particularly fluid which are particularly present in terms of the cell. So we are so when we are talking about the case of the cells, so we are having the two broad kinds of the cell. These are what? So these are the here meaning of the prokaryotes, then the eukaryotes. Now, when we are talking about what are actually the prokaryotes, in this case, what will be happening? In this case, particularly the nucleus will be present in terms of what? It will be present in terms of the inferior ratio. Often, either it will be present or either it will be in the leg ratio. Mean what? So, the particular cell which is having the leg of the nucleus or either presents in terms of the inferior amount. So, it will be coming under what case? So, it will be coming under your prokaryotic cell. Often, when we are talking about the case of the eukaryotic cell, so the cell which is having the defined margin amount of what? As well as when we are talking about the case of the plasma membrane or the cell membrane, so you are very clear about that. Means particularly the membrane which will be present in terms of the plant. So this will be particularly found with reference to what? Means your plasma membrane. These are literally being found in terms of the plant cells as well as when we are talking about the case of the animal cells. These are particularly having the leg with reference to what? With reference to these what? In the animal cells, it is having the leg of the plasma membrane or either the cell membrane. Often when we are talking about the case of the cytoplasm, so these are what? Tell me. So these are the fluid or either the viscous, yes, or either the highly viscous fluid structure or either the here mean the substances literally being present in terms of what? In terms of your cell. Often different types of the different components of the cell organelles are directly being merged with reference to what? With reference to this cytoplasm. Of course, it will be merged. Why is so? This is due to because your cell organelles are what? So these are nothing else but the nucleus. Then what? Then the nucleolus. After that, the endoplasmic reticulum. Often the Golgi complex, the Golgi apparatus. As well as what? Often you will be having the cytosol, often the vacuoles. As well as what? As well as the lamella, etc. So these are particularly being enclosed as well as coming under what? So these are literally being merged with reference to what? With reference to the cytosol. Often when we are talking about the case of the presence of the cytosol with reference to what? With reference to your cell, it will be around here many in terms of the 80% composition. So suppose that we are having the 100% of the water in terms of the ratio of the cell. After that, it will be having the fluidity mechanism. It will be employed as well as promoted in terms of the 80% with reference to what? With reference to the nothing else but these cytosol. Understood? Now, so let's understand about these. Means what are actually the different types of the vital components as well as the parts in terms of what? In terms of the endoplasmic reticulum. So these are what? Tell me. So they are the rough endoplasmic reticulum, the smooth endoplasmic reticulum, often the nucleus. And when we are talking about the case in terms of the particularly endoplasmic reticulum, so it will be having the vital types of the portion in it. Fine. So these are what? So these are the rough endoplasmic reticulum, of course. Then the smooth endoplasmic reticulum. Then what? Then you will be having the ribosomes. Often you will be having the nucleus. Fine. As well as what? As well as the region in which the protein are particularly being transported. Remember? So in this case, particularly mechanism, role, as well as the employation with reference to what? With reference to the protein is open. Will be understood now only. After that, you are often having the different types of the parts in it, such as what? Such as the nuclear envelope. Then the here, as well as you will be having the different types of the pores, as well as what? The structures in it. So these are what? So these are the nuclear membrane, then the nuclear pores. Often you will be having what? Often you will be having the ribosomes. Remember about the case of ribosomes? So what are actually the ribosomes? Tell me. So these are the smallest cell organelles. Now, when we are talking about the case of the organelles, you are very clear about that. Means what are actually the organs of organelles mean in term. Often when we are talking about who was particularly scientists who have demonstrated as well as discovered what? The ribosomes. It was discovered by whom? Tell me. So it was discovered by here in term of the Pelladi. Often these are particularly being divided in terms of the two broad categories. So these are actually what? So these are the 70S ribosomes as well as the 80S ribosomes. Remember, when we are talking about the case of the 70S ribosomes, So these are generally being found with reference to the prokaryotic cell. And you are understanding about that means what are actually the prokaryotic cell. Now let's proceed with reference to the second one. So we'll be having one, the 80s ribosomes. Often when we are talking about the case of the 70s ribosomes, so these are having the two units in terms of one larger units. Then what? Then the another one will be the smaller subunits. So your smaller subunits are particularly resembling the amount of the protein codons with reference to how much of the here units. So this will be employed with reference to the 30s. Then the larger one will be how much? So it will be around 40s. When the 40 subunits directly proportional for the larger subunits as well as the 30s is directly proportional employing for the smaller subunits. Often you will be having the proteins in it in terms of the middle structure playing important role as like the connective link employed by what? So these are particularly being attached with reference to the globular protein. Remember what are globular protein? These are soluble with reference to the water molecules or either the hydrotic molecules etc. Best example are what? So these are the pepsin, renin, often the lipase. Often you are very clear about that means what are actually these role playing important role in terms to carry out the course of the digestion. Often when we are talking about the ATS ribosomes, so these are what? Tell me. So in the case of the ATS ribosomes, so you will be having the subunits. In turn, you will be having the one larger subunits, then what? Then the smaller subunits. So the larger subunits will be around how much? Tell me. So it will be particularly here, I mean in terms of the 30. Then in term on the other hand, you will be having what? Then in the another hand, you will be having the 50. Fine. So in term, it will be promoted by what? So it will be resembling. So particularly when you add these, mean 50 plus 3, coming under what case? Or either 30, coming under what case? So it will be coming under the 80 subunits. Fine. So the smaller ones directly proportional for the 30s as well as the larger ones are directly proportional for the 50s ribosomes. Particularly being attached with reference to what? The globular protein. Coming under what case? So this will be coming under the, tell me. So this will be coming under the ATS ribosomes. Often. Now, so let's proceed with reference to this. So you could often find here on the board the resemblance of the particular structure, which is having the particular specialized what? The roles as well as the organelles as well as the components which are literally present where? Which are literally being present in terms of the endoplasmic reticulum. Find here. So here is the nuclear pores. Often you are having the rough endoplasmic reticulum. Fine. So this is somewhat the condition. So now let's understand about this which is about the endoplasmic reticulum. So these are often being further divided in terms of the two broad categories. So these are what? So these are the rough endoplasmic reticulum, then the smooth endoplasmic reticulum. Now let's understand about this which is what actually the rough endoplasmic reticulum. So in this case what will be happening? Tell me. So in this case the ribosomes will be present on the surface. Often when we are talking about this case of the ribosomes, so these ribosomes are playing important role to manufacture what? To manufacture the proteins. After that, these are particularly being transported with reference by the help of what? With reference by the help of the endoplasmic reticulum. Then by the particular influence of the other cell organelles. Remember? So we are having the here in terms of the Golgi complex, often the Golgi apparatus. Often we will be having the endoplasmic reticulum as well as the another parts in it. Now, so when we are talking about these cases in terms of the what? In terms of the rough endoplasmic reticulum, you have to understand the more things in it. So in this case what will be happening? So your rough endoplasmic reticulum is what actually? So these are like the cell organelles. So like all the other cell organelles, our rough endoplasmic reticulum are often what? So these are often the what? The cell organelles as well as when we are talking about the case of the endoplasmic reticulum. So these are what? So like all the other cell organelles, we are having the endoplasmic reticulum. In turn, we are having the rough endoplasmic reticulum. So the role is what? So these are playing important role for synthesizing the, tell me, for synthesizing the actively protein synthesis. Find here on the board here. After that, you are often having the smooth endoplasmic reticulum. So when we are talking about the smooth endoplasmic reticulum, so these are known as what? So in this case, the ribosomes will not be present on the surface. Often when we are talking about the smooth endoplasmic reticulum, so there will be particularly the synthesizing will take place often. In what mechanism? So there will be not the synthesizing mechanism will be employed with reference to the protein, but literally on the another component such as what? Such as the lipids. Often what? Often the phospholipids as well as the fat molecules. As well as what? As well as the glycogen. Now, so we are talking about these cases means what are actually the lipids. So you are very clear about that. So what are lipids? So lipids are actually what? Tell me. So the lipids are the natural organic substances which are quite a bit insoluble in water molecules or either in the H2O. Often when we are talking about these cases, so in this case, when talk the lipids play important role to provide the support with reference to the cell as well as often its different organelles as well as the different ingredients. So you could often find it on the board here. As well as when we are talking about the case in terms of what? In terms of the smooth endoplasmic reticulum. So these are known as what? So these are known as the aorta. endoplasmic reticulum. Mean in terms of what? So एवढा endoplasmic reticulum are quite bit directly proportional for what? Smooth endoplasmic reticulum. Often when we are talking about the case in terms of these examples, so this best example will be what? So these are quite bit like this, such as the, tell me, such as here many terms the testis cell, the muscle cell. So find here. Often what? Often the adipose tissue. Thus, this is somewhat the condition when we are talking about the case of the endoplasmic reticulum. So it was demonstrated as well as discovered by, it was demonstrated by the KP Porter as well as the Pelladel. Often the Calvins. Often we have understood about this in terms of the bit introductions. Means what are actually the different another components of the cells in which we have demonstrated as well as understood about what. So we have understood about the plasma membrane, the bit introduction with reference to the ribosomes as well as the nucleus as well as the nucleosomes, as well as we have often understood in brief means what are actually the here minute of the endoplasmic reticulum. Often we have understood about what? Often we have understood about the rough endoplasmic reticulum as well as the smooth endoplasmic reticulum. So you could often write these. So if you like the work with reference to the education as well as the lectures as well as what? As well as the videos. So you could often subscribe, like as well as comment below in the text. Thus I will be knowing that what concept actually you are hungry for or what haven't you understood or either got. So very thank you with reference to what? With reference to the Rahul Anand tutorials. So please like, subscribe as well as share. Thank you.